Rastatri, here we go. So continuing talking about species and speciation, I wanted to give you guys a famous case study from the 1950s of this California wildflower that used to be called Clarkia deflexa. Now it apparently has been renamed Clarkia bote. I'm not sure why. The common name for it is farewell to spring. And it's common in Southern California. It's an annual. All right, checking the chat right there. It can self-fertilize, but normally it outcrosses. And Harlan Lewis collected Clarkia from 11 different populations in Southern California and tried interbreeding them. So, you know, if we're using the biological species concept as defined by Ernst Mayr, then, you know, a species is a set of populations that are potentially capable of interbreeding with each other. So that's his original map. That's where he got his Clarkia deflexa. The range is in green, and A through K are the spots where he collected, starting, E and F is Point Conception, A and B, that's the Big Sur area, and then going around to LA. And he tried crossbreeding plants from these 11 populations. And this is the data that he got plotted as a web, where each different line pattern expresses how interfertile each pair of populations was. So a dashed line indicates that when he crossed them, he didn't even get any seeds. A narrow line is that he only got one seed. Let's colorize this. The lines that I've colored in green indicate populations that were completely interfertile all the way through the F2 generation. In other words, he crossed them. They produced hybrids that lived and were fertile, and the descendants of those hybrids were also fertile. So here we have complete interfertility, at least for 2 generations. So those are the ones that are pretty unambiguously members of the same species if we're going by Mayer's definition. Yellow, F1 was fertile, but F2 either died, was degenerate, or was sterile. So these are fertile in the F1, but the next generation shows some problems. And then in red, those lines indicate populations where the F1 generation either died or was sterile. And the thin black lines, which are all radiating out from population A, are cases where no F1 generation was even produced. So you can see we've got a range of conditions from absolute hybrid, absolutely no interbreeding, where, you know, hybrids don't even produce seeds, to complete interfertility up to at least 2 generations. That's those green lines. And then some conditions that are in between. And if you look at this, you could maybe make a case that population A should be a separate species because it could not even produce seeds with any of the other populations that it was tested with. But notice that, for example, D is fully interfertile with I, and D is fully interfertile with E, but I and E are sterile with each other. The F1 generation either died or was sterile when you cross I and E. And you can do lots of these things. Let me see. C and, I and C are infertile, I and F are infertile, but C and F are fertile. There's no way you can really divide up that diagram to give you clusters of populations that are and aren't consistently reproductively isolated from each other. It's aside from maybe cutting out A, because that doesn't seem to be fertile with anybody. You get all kinds of in-between cases between complete interfertility and complete sterility. And exactly how to divide these up just is not immediately obvious. To try to alleviate some of the difficulties of the biological species concept, there's been a number of alternative species concepts that have been proposed. There's something like a dozen that are out there. I'm not even talking about all of them. But these have problems of their own. We've talked about the biological species concept already. The species definition that's commonly used in practice is the morphological species concept, which defines a species by the unique physical features it's got. This is the one that's used implicitly in all of your field guides. All of your bird books that give you the difference between red-tailed hawks, red-shouldered hawks, and Cooper's hawks. Or all of your plant guides that give you the distinction between, smooth sumac and winged sumac, or whatever the case might be. This is what you may have used before if you've done field biology classes like ornithology or systematic botany or anything like that. The ecological species concept proposed by a gent at U of Chicago named Lee Van Valen basically defines a species by the niche that it inhabits. So no matter how different two populations might look, whether or not those populations can interbreed, if they share a niche and neither one drives the other one out, we'd call them the same species. We'd simply, instead of saying that which a species occupies is its niche, we'd say a species is whatever it is that occupies what we're calling a niche. And then the behavioral species concept focuses on defining a species by what they call its SMRS, that's specific mate recognition strategy. Most species have some method of recognizing potential mates. We don't always see what they are because a lot of species use senses that are not the senses that we are geared up to focus on. There's a lot of moths, for example, that you can hardly tell apart even by careful examination, but the moths can tell themselves apart perfectly well because they're not using sight to find mates. They're using smell, and they're extremely sensitive to very subtle distinctions in pheromones. So that would be an example of a specific mate recognition strategy that humans just aren't that good at recognizing because our sense of smell is not nearly as good as a moth's. The problem is that they don't always give you consistent answers. And here I'll go back to an even earlier classic case study of eastern toads. He went around collecting toads from all over the eastern United States and collected, I won't go into all of his findings, this is just a small part of it, but he looked at two American toad species that overlap in their distribution, Bufo americanus and Bufo fowleri. So there's what they look like. And in the part of his findings that I'm going to show you here, he collected female Bufo fowleri from Indiana and tried crossing female fowleri from Indiana with male Bufo americanus from elsewhere. And here's where he got. All of the females here were from Indiana. So if he crossed males from Bloomington, Indiana with females from Bloomington, Indiana, he got anywhere from 40 to 95% of the eggs yielding tadpoles. If the males were from Winona Lake, Indiana, he got nothing. And if he collected males from Tulsa and mated them with females from Bloomington, Indiana, he got everything from 4% eggs with tadpoles to 95%. So everything from almost complete intersterility to virtually complete interfertility. You can see these numbers are falling out on a continuum. You know, some crosses are more fertile than others, but there's nowhere you can absolutely say, okay, these two cannot interbreed, except maybe the Winona Lake one. So are these separate species? Behaviorally, probably so. They breed in different habitats and the mating calls are different. Bufo fowleri is said to drone, where Bufo americanus is said to trill. Morphologically, they're probably not separate species. They're not consistent enough that you can tell them apart just by looking. Although there's been quite a bit of debate on the matter among people who really get into toads. If any of you have taken herpetology, you might get some insight into this, but apparently they're not different enough morphologically that people are consistent on whether they should be called separate species. Ecologically, they overlap so much they probably aren't separate species. Biological species concept, do you remember that important scientific principle from the last lecture? The principle of, the principle that we don't get neat answers. There are cases where if we ask if two populations are one species or two, either a yes or a no answer is the wrong answer. And all we can say is, and that's okay. Because as I've said, this is what we would expect on a world where biological evolution is happening. Species ought to be fuzzy. Species ought to have a lot of in-between cases and I'm not sure cases, and mostly they're separate, but occasionally genes from one introgress into the other, or mostly they're the same, but maybe kind of not, sort of, but maybe. We ought to have cases like that. And in this particular case, we'd probably call North American Bufo a species complex, meaning that we have a lot of populations that are sort of separate species, but not entirely, but maybe, but kind of not, but sort of. Hope you followed my technical jargon there. And this sort of thing happens all the time. There are lots of cases where we just kind of have to throw up our hands and say, okay, it's a species complex, but we really don't have unambiguous places to divide it into good, well-defined, separate species. And again, that fuzziness is what we would expect in an evolving world. There should always be cases of species coming into existence but not fully separated, and it doesn't even matter what species concept you're using. And Ernst Mayr, in his later career, after he had gotten back from New Guinea and ended up as a professor at Harvard, becoming really one of the grand old men of biology, became a historian of biology, and in a very thick book called The Growth of Biological Thought, argued that one of the most important events in the history of biological thought was a shift from what he calls typological thinking to population thinking. What's that? So here's Carl Linnaeus, a Swedish physician with an ego that would not fit through a barn door, who found himself on a divine mission to understand the entire natural world. Like the Blues Brothers, he was on a mission from God. And this is Linnaeus talking about himself in the third person, which is a little creepy, saying God has allowed him to see more of his created work than any mortal before him. God has endowed him with the greatest insight into natural knowledge, greater than any has ever gained. Basically, low self-esteem was not a problem Linnaeus ever really suffered from. And he was the one who'd figured out, you saw him, and you saw this mentioned briefly in the Day the Universe Changed video that you might remember from back at the beginning of the course. Linnaeus figured that if you could just figure out the right way to classify all of life, you could get inside God's head, and you could understand, at least as much as a human ever could understand, what God had been thinking, what plan he had when he'd created everything on October the 26th, 4004 BC at 9 o'clock in the morning. And he went back to Aristotle, and it was Aristotle who'd said, okay, if you want to classify things, first you have to define what makes the group alike, and then you have to define what makes each member of the group different from the others in the group. And Aristotle's names, at least in Latin translation, for these things were genus and species. That what makes a group, what makes group members like each other is their genus, and what makes each member of a group different from each other is its species. And these meanings still survive in words like generic and specific. If I say that somebody's just a generic college student, I mean that that person has all of the features of college students, but nothing that really distinguishes him from any other college student. You know, just your basic, you know, ramen eating, sleep deprived, caffeinated, twitchy, generic undergrad. Okay, I don't know if that really has any meaning, but that's what generic originally meant. It meant having all of the features of your type, but the implication is it doesn't have anything specific to itself. So here's Aristotle. His genus is humanity, man, because all men have 2 eyes and a nose and 2 ears and all of your, you know, bog standard features that you see. His species is Aristotle. He's a specific member of the genus because he's got a bald spot, a beard, a big nose, goes around philosophizing, once got run out of Athens for philosophizing too much, used to serve as a tutor to Alexander the Great, founded political philosophy and also dissected octopuses. Yeah, all that. Linnaeus, of course, takes that ball and starts running with it, and of course, formalizes the use of genus and species in biology. So that, by the way, is a book he wrote called Systema Naturae. You can see it's open to the page on the order primates, which he defines as dentes primores superiores quatuor paralleli mammae pectorales binae. Front upper teeth, that's your incisors, 4 and in parallel, nipples on the chest and paired. So yes, primates are defined by having 4 upper incisors and 2 nipples on the chest, instead of rows of nipples or udders or something like that. And then below it, that's his definition of genus Homo, which is humanity. And his definition is just nosce te ipsum, recognize yourself, or I'm talking about you. And he defines two species, Homo diurnus, which is us. He would later change this to Homo sapiens, by the way, and Homo nocturnus, way down below. Here's Homo diurnus, which would later be changed to Homo sapiens. And then Homo nocturnus was his name for the orangutan, which wasn't very well known at the time. Okay, that's from, I think, the 7th edition of his book. The 10th edition came out in 1758, and that was the first edition, at least for animals, that used binomial names consistently. There were scientists before Linnaeus that tried to name species in various ways. We don't use any of their names now. The oldest Latin names for species that are still in use are the ones in the 10th edition of Systema Naturae. And Linnaeus coined a lot of the ones for very common animals: Homo sapiens, Bos taurus, the cow, Sus scrofa, the domestic pig, Canis familiaris, the domestic dog. A lot of Linnaeus's old names are in fact still in use. However, Linnaeus never really doubted the biblical creation story. The Arkansas State Legislature would love it. He considered that each species must have been created separately by God. He did later suggest, based on some of his studies of plants, that you could have new species appearing by hybridization. You could cross two species and get a third species that wasn't like either parent. And as we'll see, there are cases where that actually happens. I'm going to talk about that next week, in fact. But that's still not really creating anything new. God sort of deals the cards, and then hybridization, you can shuffle them, but you're not putting anything new into the deck, right? You can shuffle the cards and deal out great diversity of poker hands, for example, but no matter how many times you shuffle the cards, you're never going to get the cards to produce a 14 of spades. Or, we've got a king of hearts, a queen of hearts, a jack of hearts, but you're never going to get it to produce a plumber of hearts or an accountant of hearts, or a small-time blogger and media influencer of hearts. You're never really going to get anything new by hybridization. So for Linnaeus, every species is a separate creation with maybe some limited hybridization, but still bounded within very strict limits. Since he wanted to get inside God's head, he defined each species by some sort of intrinsic essence, some sort of nature that God had defined, the blueprint that existed in God's mind before God went around and created everything. And that essence couldn't really change. As he put it, the invariability of species is the condition for order in nature. If species do vary indefinitely, then creation is going to look like a complete mess. But no, there's got to be some sort of fundamental plan of every species. And individuals might vary, but that variation is only an obstacle for understanding what that essence is. So there you've got an extremely variable species right there. That's the Cuban land snail, Polymita. Thank you, Aaron. And you can see it's quite a variable species. You've got lots of different colors and banding patterns and even proportions of the shell. But for somebody who's working in Linnaeus's mindset, that variation is basically irrelevant. it's only going to get in the way. What's important for you to do, if you're a biologist, is to understand that unchanging nature, that essence, that thinginess that every species has. Focus on what all members have in common rather than this variability. We still use this in biology when we define new species. If you go out and you're exploring somewhere and you find an animal or a plant that doesn't look familiar, you take some back to your lab and you go through all of your scientific papers, which in the old days would be in great big file cabinets. These days are probably all PDFs. And you carefully compare your specimens of what might be a new species with all of the other similar species that might be out there. And if it still looks like it's new, well, these days you'd probably get DNA sequences as well. But at least up until quite recently, you would compare your specimens to all other similar species. And if they seem to be consistently different, you would describe your species. You'd write up a paper saying a new species of such and such from so-and-so, a new species of blister beetle from the rainforest of Papua New Guinea, or something like that. There's hundreds of thousands of scientific papers with titles that look like that. And you'd write up a formal description of what the new species looked like and why it was different from all other species that it might be confused with. And you'd present, where you got it and what habitat it was in and so on. But to make that name valid, you had to take a type specimen and deposit it in an established museum. Now, the museums where these things go aren't necessarily display museums. Sometimes they are. The Smithsonian, for example, has huge exhibits, but sometimes they're not. There is a major museum of entomology on the University of California at Berkeley campus, the Essig Museum of Entomology, but people don't know about it because, at least when I was there, it had no public exhibits at all. Entertaining the public wasn't what it was there for. What it did was maintain collections of insects, including type specimens like this. And if you were studying California insects and you found something you thought might be new, ultimately you might have to go to the Essig Museum and compare your specimens against the type specimens, because the type is supposed to be a permanent standard reference for defining exactly what that species is. This is a type specimen of a Brazilian flower kept at the Botanical Gardens at Kew in South London. And if you were studying Brazilian flowers and you found some specimens that might be this and might not, or if you were studying Brazilian flowers and you said, hey, I don't think this species is really different, I think it's just a variety of another species, ultimately your arguments go back to the type specimens, because the type specimen is basically a permanent record of what you think that species means. It used to be, this is now changed, but a long time ago, the international official definition of the meter was the distance between two very thin lines engraved on a bar of platinum alloy kept in Paris. And that was the standard for what the meter was. That's very specific. And all of your you know, maybe not for the average yardsticks that you could buy at the hardware store, but for precise scientific purposes, ultimately all your definitions of the meter went back to that 1 bar of platinum alloy in Paris.
It actually goes to the speed of light.
Yes, Aaron, what we have now done is come up with a better way to define the meter that does not depend on being able to go to Paris and get access to 1 bar of metal.
Yeah, it's the distance that it's 0.c, or it's the distance that light would travel in 0.c seconds.
Sounds good. All right.
That's that's the way you define it.
Okay, right. And the way we used to define species was the same. That ultimately, if there was any doubt as to what a species meant, you could go back to the type specimen, and that would be the permanent standard of reference for what that species was. And typological thinking like that, that's what I mean by typological thinking. A species is defined as being a reflection of some kind of permanently definable type. And this is very explicit in the creation science movement. This is from the Statement of Belief of the Creation Research Society, based out in that buckle on the Bible Belt known as San Diego, California. They get a little bit annoyed if you ask them to pin it down, but if we look at this, all basic types of living things were made by direct creative acts of God during the creation week described in Genesis. Whatever biological changes have occurred since creation week have accomplished only changes within the original created kinds. Now, trying to get them to come up with a real definition of what a kind is kind of like, I've tried to pin them down on the point, and it's kind of like nailing Jello to the ceiling. But the idea is that all living things you can put in pigeonholes, in boxes. Maybe there's been some change and there's some variation, but it only happens within very strictly defined limits. That God's original creation established the fundamental types of living things that there are, and you cannot get change that crosses any of those boundaries between different kinds of living thing. If you're working with that sort of mindset, classifying species is a lot like classifying atoms. If you think about it, you know, we have a scheme for classifying atoms. It's called the periodic table. And in that scheme, every atom is classified by the number of protons in the nucleus, right? So if you have 1 proton in the nucleus, you're hydrogen, 2 and your helium, 3 and your lithium, 4 in your beryllium, ...
Yeah, also the amount of electrons.
Yes, the electrons in the outer shell should balance the protons in the nucleus, but sometimes it doesn't, and then you've got electrical charge. I'm coming to that. Aaron, let me do the teaching here. Okay, I know you're bored. So you can classify the elements based on the atomic number, protons in the nucleus, which, as my kid was saying, should balance the number of electrons in the shells, but if it doesn't, you have an ion. And you can further subdivide by the number of neutrons in the nucleus. So we have hydrogen, which can exist in one of 3 isotopes, depending on whether you've got 0, 1, or 2 neutrons, protium, deuterium, or tritium. Right. The reason I brought all of that up is that that's the kind of pigeonhole classification that's typical of typological thinking. Thank you. You're never going to get a case where you have a bunch of atoms that have something between 6 and 7 protons in the nucleus, right? You can't have 0.37 of a proton in the nucleus. You're never going to get an atom that's kind of like it might be carbon and maybe could be nitrogen, but well, I don't really know. You're never going to get an atom with an atomic number of 6.327. right?
Unless
You're
manage
either going to
select
have
and
6
make
or
atoms.
7. And if you split elementary particles, things get weird, but you don't really have atoms anymore.
I know, but if you split the actual atom in half and then figure out how to make it orbit
Okay.
like an electron, that it could I don't know.
Thank you, but could I do the teaching here?
Yes.
Thanks. They're paying me. They're not paying you, so don't give them free labor. Hi, Cat. And here's why biological classification now is fundamentally different. The whole driving force of natural selection is variation. It's variability. Species can change, they do change, and variation as acted upon by natural selection is what makes that change possible in the first place. You know, you can't have atoms that are maybe carbon and maybe nitrogen, but we're not sure. But you certainly can get toads or lizards or farewell to spring flowers that are maybe this and maybe that, but we're not sure. It happens all the time. Sometimes that variability slops over the edges of the nice, neat, tidy little definitions that you'd like to be able to draw. So this just shows a moth from Arizona that you can see is variable in its color and pattern. Darwin went so far as to suggest that species should not be considered to objectively exist at all. Species would be a term of convenience, you know, because it's useful to be able to talk about live oaks or farewell to spring flowers or American toads or what have you. It's useful to talk about them, but we're not really referring to anything that's really out there. Kind of like the way in species would be more like classifying colors instead of classifying atoms. Because, you know, we give names to different parts of the color spectrum, you know, red, orange, yellow, green, blue, indigo, violet. But really there's a continuum from red to violet, and there's all sorts of colors that are in between. And it's just for convenience sake that we divide up the color spectrum the way that we do. And in fact, different colors divide it up differently. Choctaw, for example, does not have different words for green and blue. Russian has separate words for what we'd call light blue and dark blue. Different cultures divide that up differently. And dividing up species is the same. It's convenient, but we're not pointing to anything that's absolutely unambiguously real. So as Darwin puts it, the only distinction between species and well-marked varieties is that the latter are known or believed to be connected at the present day by intermediate gradations, whereas species were formerly thus connected. In short, we shall have to treat species in the same manner as those naturalists treat genera who admit that genera are merely artificial combinations made for convenience. This may not be a cheering prospect, but we shall at least be freed from the vain search for the undiscovered and undiscoverable essence of the term species. So don't worry about trying to define what a species is. What's real is the variation. That's what's important. To Linnaeus, variation gets in the way of understanding the species' essence. To an evolutionary biologist, now, there is no essence. Variation is all you've got. And Ernst Mayr has emphasized the importance of that shift. Instead of thinking of life and species as being defined by these types, we think of species as sets of populations, and those populations are constantly in flux. No two members are alike. You know, allele frequencies and trait frequencies can constantly be shifting, and populations vary, much like these Donax variabilis surf clam shells from the Gulf Coast of Florida. You can see they're all variable in color and pattern. And a modern person who realized the importance of this was a fellow by the name of Alfred Kinsey. Here he is. Kinsey was a specialist on a group of small stingless wasps in the genus Cynips and related genera. And Cynips are oak gall wasps. Cynips adults will lay eggs. Ow, sorry, the cat is trying to stab me. Hi, cat. Gall wasps in the genus Cynips will lay eggs in the tissues of almost always oak trees. There's a few that might lay eggs in closely related species like chinkapins, but it's almost always oaks. And the eggs will hatch into larvae. The larvae stimulate the, hang on, somebody's trying to get in. The larvae will stimulate the cells of the oak to start growing and dividing and creating a swelling. And then the larvae will eat the swelling from the inside and eventually pop out and go on to mate and lay their eggs in more oaks, and round goes the circle of life. So there's a cynipid wasp right there. You can, if you look at your average oak tree in summer, you'll find cynipid galls. They can be on the twigs, they can be on the leaves. Sometimes they can be small and kind of cone-shaped. There's galls that I see in the fall in the nature reserve that are about the size of ping pong balls, and you find them on dead leaves in the leaf litter. And by the time you find them there, they're pretty much hollow, and the wasps have flown away a long time ago. They call them oak apples sometimes. And I've spotted those in the nature reserve. If you poke around enough in there, you'll see the same as I. Kinsey, his work on gall wasps isn't perhaps not that well known, but he really, really wanted to get to what it is that a species really is. And he realized that in a lot of cases, species of insect had been defined by somebody goes out and collects a couple of insects, finds that they look different from others, and names a new species without looking at the variation, without looking at without even trying to answer the question, are these, what's the variation in my species? Have I really found a new species, or have I found, something that's part of the range of variation of an established species? Well, nobody was really looking at what the range of variation is at all. So Kinsey and his students would drive all over the United States and ended up collecting over 71,000 cynipid specimens in an attempt to try to find, just how much variation there was among cynipid species. As Kinsey put it, too many systematists attain their objectives when each species is represented by a half dozen specimens pinned in their cabinets. These are the systematists responsible for the definition of systematic entomology as the science of transferring pins from one box to another. You may not realize it, but that's actually a pretty sick bird. So instead of trying to base his understanding of gall wasp species just on the basis of a half dozen specimens pinned in a box, Kinsey surveyed cynips from all over the United States, built up a huge collection, and concluded that species were capable of seemingly limitless change. They could vary, and they could blend into each other, but very often you could define stable entities that did seem to fit the bill of what you might want a species to be. He compares a species to being like a stream. If you follow a stream, it changes endlessly. Other streams can flow into it. The stream might divide and fork. Sometimes the stream might go through a swampy patch, and if you're in a very muddy, swampy area, it might be kind of hard to figure out where the stream actually is because it's all just a bunch of mud or something. But yet it was still useful to talk about streams. You can still talk about streams even though the streams are not fixed and stable and always flowing in the same channels. Streams can join, streams can separate, streams can get very swampy. Most of the time, it can be useful to define streams, even if you can't always pin down exactly what you mean. Oh, almost forgot. I wanted to say something else about Kinsey. He's famous today, but not for his work on gall wasps. Back in his time, back in the 20s, he was a professor at Indiana. And they were still doing this at Tulane University when I was an undergrad. They haven't done it here for as long as I've been here. But a lot of universities used to teach a course in, they'd usually call it something like marriage and the family, but it was basically sex education. And like many academic biologists, sometimes we have to teach non-majors courses in service to the university. And Kinsey had to teach a portion of this marriage and the family course at Indiana U. And being a conscientious professor, Kinsey did some research and found that at the time in the 20s, there were all kinds of descriptions that had been published of what was considered abnormal sexual behavior. Of course, our definitions of abnormal have shifted a bit, but that's another story. But there were all sorts of descriptions that were published of, sex criminals and, flashers and fetishists and all sorts of things like that. But nobody had any real data on what regular, ordinary people actually did. We knew a lot about, again, what would have been considered abnormal at the time. A lot of papers had been published on that, mostly published in Latin so that uneducated people couldn't read them and get any bad ideas. But no real studies of what the baseline was. How can you define abnormal if you don't know what's normal? Kinsey went on to send out questionnaires to people all over the United States. And just as he collected 71,000 gall wasps to try to figure out what species might actually be, he sent out tens of thousands of questionnaires for the same reason, to find out what is the range of variation in human sexuality. What are the forms that it comes in? And he published his results in a couple of books, Sexual Behavior in the Human Male and Sexual Behavior in the Human Female in, I believe, 1953 and 1955. He got a lot of fame for this that he might not have wanted. In fact, when Sexual Behavior in the Human Female came out, he was denounced by irate representatives in the US Congress who tried to defund his work and threatened Indiana University with all sorts of horrible things if they didn't cancel it. Well, fire him. They talked that was just firing back then. Because he'd found out a lot of things that people didn't really like to hear. One is that premarital sex was, for all that everybody refused to talk about it, it was actually pretty common back in the 1950s.
Still is.
Well, yeah, we talk about it more. In the 1950s, there was...
Still happens.
I know, hush.
About the same amount.
Yeah. In the 1950s, though, you know, people like to keep up the fiction that good girls didn't. But in fact, good girls did. The frequency is not much different from what it is today. You know, your grandma quite possibly got around quite a bit. They just didn't talk about it so much. Kinsey found, we've revised this statistic a little bit because the answer you get depends very much on how you phrase the question. There are some aspects of Kinsey's questionnaire that have had to be improved, but Kinsey found that roughly 1 in 10 of human males were homosexual, or at least had had homosexual experience. Kinsey concluded, his joke was that half the population of Indiana should be in prison and the other half was anemic. Half of them had done something that should have gotten them in prison by the laws of the time, and the other half would have, except they didn't have the energy, was his joke. And it made him a notorious figure, especially on the far right of the time. There were claims he was trying to undermine the moral fiber of America and the purity of our women and all this. But he persisted. The Kinsey Institute for Human Sexuality is still going strong at the University of Indiana. And although we've had to make a lot of, you know, updates and modifications to his findings, you know, because again, you know, statistical methods have changed and so have ways of writing surveys. You know, we're more careful about writing leading questions and things like that. Nonetheless, he pretty much founded the field, almost without meaning to, of academic sexology, of studying human sexual behavior as another form of human behavior, not to try to, you know, point fingers and giggle over what's abnormal, but just to figure out what normality is in the first place, and using large data sets in order to do that. Instead of picking out a couple of weirdos, you survey the entire range of variation. And it's the same basic type of thinking that informed his research into these gall wasps. There's actually a good essay on Kinsey's work by Stephen Jay Gould called Of Wasps and Wasps. Don't know if you get it, but wasps as in gall wasps and wasps as in white Anglo-Saxon Protestants, the group that Kinsey surveyed the most. And his studies on both relied on the same insight of the need to fully quantify the range of variation we find, because variation is fundamental, because variation is not something that gets in your way of perceiving the ultimate truth, the way it was for Linnaeus. Variation is the ultimate truth. And that's what drives evolution. That's what we focus on, rather than focusing on typological thinking. I guess in the field of human sociology, we do the same. Instead of dividing people into good people and those gays and bad girls and things like that, we try to look at the total range, look at all of the variation, and try to understand that on its own terms. And that strikes me as a more humane way of doing research into the human condition. But I'll get off my soapbox here and stop the recording, leave you with these thoughts.